Dr. Faraz Malik. He's assistant professor of bioscience at COMSATS, Institute of Information Technology in Islamabad, in Pakistan. Dr. Malik received his PhD from CIIT in Pakistan. He also holds a master from the National Center of Excellence in Molecular Biology at the University of Punjab, and is currently an HEC postdoctoral fellow researching in metastasis and angiogenesis. And his talk is entitled Role as SEMA3C in Breast Cancer Progression and its Effects on Cancer Cell Proliferation, Adhesion, and Invasions. Dr. Malik.
My name is Faraz Malik, and the portion of the research work which I am going to discuss with you guys today is about the role of semaphorin 3C in breast cancer progression and its effects on cancer cell proliferation, invasion, and adhesiveness. This work was done at Metastasis Angiogenesis Research Group, Institute of Cancer and Genetics, School of Medicine, University of Cardiff. And before I begin my talk, I would like to briefly introduce you about the semaphorins and revise myself as well about the semaphorins. The semaphorins are the molecules. Their potential involvement earlier was identified as neural growth axon guidance cues, but later on it was identified that these semaphorins are also involved in playing a pivotal role in several other organs. And the basic structure of semaphorins, it contains a SEMA domain. From PSI, it stands for plexilin, the receptor for semaphorins, in most cases, semaphorin integrins domain, and a basic C-terminal domain for the majority of the semaphorin molecules. On the basis of this structure and sequence similarities, the semaphorins are categorized into 8 classes. Class 1, 2, and 5 belong to invertebrates, while class 5 for viruses. The rest of the classes belong to the vertebrate family. And I am talking about semaphorin 3C, so it belongs to the class 3, and there are 9 members, out of which I am discussing about semaphorin 3C molecule. So as far as the semaphorin 3 is concerned, its binding receptor is neuropilins, neuropilin 1 and neuropilin 2. Its expression in both neural as well as non-neural organs has been observed, and it has been reported in several other cancers like lung, ovarian, and prostate cancer. But the studies regarding the breast cancer cohort is, and you can say it's a limitedly addressed domain so far. And the problem with this molecule was the pleiotropic mediator of development and diseases, that in some cancers, the expression profile of this molecule is upregulated, while in certain cancers, the molecule is behaving quite differently. So in my present study, what I'm going to show you is the expression profile of semaphorins in the breast cancer cohort and its effects on semaphorin 3C on cancer cell lines. So briefly about the methodology. For breast cancer cohort, from breast cancer cohort, I isolated RNA and synthesized cDNA from the biopsy tissues and used normal background tissue as control, and analyzed the expression profile of semaphorin 3C using real-time PCR technique. And then the expression profile of this molecule was correlated with the clinical features we have about the cohort data with us. And that was the portion of the clinical trial we did, clinical study we did. And for cancer cell lines, I was interested to know how this molecule is actually behaving in vitro model. And I want to study the cell proliferation, adhesive response, and invasion under the influence of semaphorin 3C knockout cell lines. And for that purpose, I generated semaphorin 3C specified small stretches ribozymes and then transfected those ribozymes in the cancer cell lines. And after successful transfection and successful confirmation of these cell lines, I studied the characteristics of semaphorin 3C on cell proliferation, adhesiveness, and invasion. And this model can later be extrapolated for in vivo design studies. So my results were, for the clinical trials, in grading sample, I find a strong, significant correlation in advanced advanced grades like in poorly and moderately differentiated tumor samples, the expression of semaphorin 3 was higher. I haven't observed any significant correlation with TNM staging and Nottingham prognostic index values of semaphorin 3C with the cohort data. However, the data over here, that's another way of presenting my findings. That the graphical view is showing that the semaphorin 3 expression was increased in poorly differentiated or grade 3 tumor samples. On the A portion, I am studying the whole cohort details, while on the B side, I am presenting you the ductal carcinoma findings what I have. Semaphorin's correlation with the estrogen receptors is very, what you can say, a controversial domain so far. Some of the studies report that the semaphorins are largely, by and large, regulated by estrogen receptors. But in my study, I have observed estrogen receptor alpha positive patients have a strong reduction in semaphorin 3C expression, while there was no correlation with estrogen receptor beta positive patients are negative. And for the cancer cell lines, what I observed and what I did, this picture is actually presenting a successful knockout of MDA231 and MCF7 semaphorin 3C expression. In 1 and 2, both the cells lines were expressing strong expression of MDA231 and MCF7 breast cancer cell lines were showing semaphorin 3C expression. After transfection with ribozyme, the expression of semaphorin 3 was lost, and these cell lines were used or compared with the normal controls in which the empty vector, I use PAF6 vector as a normal control or a mock cell line to study the effects of cell proliferation, adhesiveness, and invasion. Briefly about the growth assay. For the growth assay, what I did, I used a 96-well plate, most of the audience know, and the cells were suspended, like 3,000 cells I suspended in the normal medium, and I used 3 sets of plates, fixed them on day 1, day 3, and day 5, and I want to compare whether the cell proliferation is somehow influenced by semaphorin 3 expression or not. And for this purpose, after the 5th day, I observed that there is a reduction in the growth or the cell proliferation. And when I compared my MCF controls with MDAs, and similarly with MDA231 control cell line is showing increased proliferation when compared with the knockout cells. So from here, I excluded one result, that the reduced levels of semaphorin leads to reduced cancer cell line proliferation. In the second part, effect of semaphorin 3C on cancer cells attachment. Over here, I'm trying to show you is the 96-well plate, and this plate was initially coated by a matrigel. Matrigel is basically a replacement for the basement membrane we know in vivo models, but for in vitro studies, we use matrigel, a commercially available product. And we initially coated our 96-well plate with this matrigel, and then I seeded my cells for a very brief time interval. The idea behind this assay was to study the influence of cancer cells attachment with the basement membrane. And I am not studying over here the homotypic interactions with different cells. I'm studying over here the cells attachment with the basement membrane, so that how much the cells get strongly adhered or weakly adhered to this. So after a brief incubation at 37 degrees centigrade, like I said, it's around 40 minutes, I removed the medium, flicked my plate, and washed that with BSS solution and stained it with crystal violet. Observe these cells, count them under microscope, and after counting, I presented this data to you, that in MCF7 knockout cell line for MDH231 knockout cell lines, the expression of semaphorin 3C is responsible for the reduction of attachment of cancer cells with the basement membrane. And I repeated this assay several times before giving you a conclusive result in this, and this is a significant correlation. And the pictures which I am presenting to you is like if you can see from the leftmost side is MCF7's control cells. The next cells are the knockout semaphorin 3C expression cell line. And the number of cells you can clearly visualize are reduced in knockout semaphorenes. The third essay was about the invasiveness. A basic principle of Biodon chamber was used, and on my right upper corner of the slide there is an insert which I used. In this insert I initially coated with the matrigel, the same gel, the same matrigel which I used for adhesion assay with a variation in the concentration. And after that, I seeded my 45,000 cells or 15,000. It ranged very depending on my research work over here. I would like to acknowledge all the patients who participated in this study. I would like to extend my gratitude to Higher Education Commission of Pakistan for supporting my research work, and COMSATS for facilitating me this, or Cardiff University not least, for hosting my stay for this research. Thank to the organizers of this conference and your valued time to listen to me so patiently. Thank you very much.
Thank you for your talk. Do we have any questions?
Okay, so this is regarding the inserts. Well, so we use this model or this technique to capture two types of cells separate from each other. So the cells in the insert are not able to penetrate and go beneath, right? So it's a different thing.
So Dr. asked a very interesting question about the cells discrimination. You're talking about how to differentiate the two cells, if I'm understanding it correctly. Can we go back to, can I go back to my previous slide or some more? All right. Actually, what I did was I seeded my cells on the top of the insert. So I only seeded my cells inside the insert, not in the 26 well plate. Okay, so it was like for the keeping of these cells on the top of the insert and leaving these cells inside the insert with a matrigel already coated, and left this setup for 3 days. In 3 days, the cells actually travel from these pores into the other side or the bottom side of the insert, and I counted those cells. So, yeah. So, yes. So if you see, I initially coated my matrigel on over here, put my cells in this insert. Now the normal medium and all those cells are over here. Put this insert in this 24 well plate, okay, and leave this plate for 3 days. So the idea behind is that the cells are going to travel and pass from these pores over here to the normal medium. That's the invasiveness property of the cell. And over here, if you can see, these are the cells which are present on the other side or on the lower side. If I invert this insert, I counted the cells from this bottom, so that how many cells actually invaded that matrigel and crossed those pores, okay? And these are the pores of the matrigel from where the cells actually invaded and crosses. So that's the Biodon chamber presentation means. And there are other assays you can do. I mean to say, if you are interested, you can introduce certain chemicals on the top of the normal medium and study how the cell behaves in terms of any hormone or in terms of any drug you want to design, that how the invasive property could be regulated under the influence of this particular chemical or compound. Okay, so I hope.
Right. Do we have any other questions? All right.
Yes.
Thank you, Dr. Malik.
Thank you.